Hi, welcome back, and we're in our third part of chapter 27 on prokaryotes. In this final section, I'd like to go over metabolism. So we're going to talk about how bacteria obtain their energy in carbon sources, how they interact with oxygen in the environment, and there's a variety of substances or minerals we can look at, but we're focusing on a very important one, nitrogen. Then I'm going to talk about the phylogeny, how the archaebacteria and eukaryotes relate to each other, and what evidence we have for that. And finally, I'm going to move on to talk about some things that aren't in chapter 27, but I'd like to kind of round up our picture of life, this early part of life, about what we call non-living bioparticles. And that would be viruses, viroids, and prions. And then I'll finish up with a few disease terms that are related to bacteria and viruses and things like that. And they'll be like antiseptic and antibiotic. Okay, so let's get going here with that. And so I'm going to go over metabolism, three domains and other forms of life. And the three types of metabolism. So metabolism is breaking down and building up substances inside the cell. And so the things we're looking at is how do these bacteria and archaea get their energy, and how do they build carbon-carbon bonds, like to make that peptidoglycan in the cell wall in bacteria. How are they going to do that? And then we'll look at, there's a variety of elements we can look at, but we're going to focus in on oxygen and nitrogen. And so looking at the major modes of nutrition, looking at energy, we know of one type of energy source, and that's in plants, and that is photosynthesis. Don't know why it's doing that. And the chemistry of it you learn in 130. But to put it into a very simple pattern, it's a two-step process. In the first process, it uses sunlight to make ATP, which is a chemical energy source that the cell can use. And in the second step, it uses the ATP, it being plants, to make sugar, which is a carbon-6 compound. And so basically, it's taking in carbon dioxide from the atmosphere and trapping it into glucose. Okay, so this is what happens with plants, right? So it's a two-step process called photosynthesis. And so if we were to take and divide these in two, we have one that has to do with energy and one having to do with carbon. And in eukaryotes, all eukaryotes are doing what plants do, and they're derived off of that plant chloroplast, whether it be a seaweed, a brown algae, or a red algae, or a dinoflagellate, or a diatom. They're all doing this. Right? And it's a complete process where they're going through both steps, and so we kind of put it all together. But when we're looking at bacteria, we have some that just do this energy process. They make ATP, but they can't take in carbon dioxide, so they have to eat food in the environment like we do, right, to build their carbon-carbon bonds. They can't take it from the atmosphere. Then you have other ones that can take in carbon dioxide from the atmosphere and using ATP, but they have to do something else to make the ATP because they can't use sunlight. And so we're going to see that in the prokaryotes, it's more complicated. We have more options. There's more diversity than what we see in the eukaryotes. So first step is energy source. How do you make your ATP? And if you make it from sunlight, that would be a phototroph. If you have to eat something to make your ATP, that's energy. It's a chemotroph. You're using chemical energy. You're breaking something down. You're taking in sugar and degrading it to make ATP. Second thing is if it gets its carbon from the atmosphere and it makes sugar or makes peptide and glycan, it's auto. It's doing itself. Troph is feeding. So it's feeding itself. Right? If it's a heterotroph, right, it's requiring the carbon in particular to come in already adjoined. It's not from the atmosphere. It's breaking down the cell wall of another cell. It's breaking down fat. It's doing something, but it's taking in food energy, not carbon dioxide from the atmosphere. And so if we put terms with each of these, we can see we can get more combinations than exist, right? Because if in eukaryotes, because we look at just eukaryotes, all we have are plants, which would be a photo, autotroph, and then we have animals like us. We're a chemo, heterotroph. Plants use sunlight to make their ATP, and they use carbon dioxide from the atmosphere to make their sugar. We have to eat in other things to get our energy, and we have to eat in other things to build our materials, right? So to assemble the protein, we need to have protein in our diet, and then we reassemble it into human protein. Okay, so if we look, we have 4 different compounds, right? And so photoautotrophs would be similar to plants. Chemoheterotrophs would be similar to animals. But putting it in bacterial terms, this would be something like E. coli, which we used in lab. These would be all of these cyanobacteria, which we'll look at in lab. And then you have photoheterotrophs. Those are pretty rare. There are a few archaebacteria in ocean and saline environments. Chemoautotrophs are actually really important in deep sea vent. So in the bottom of the ocean, in the deepest parts where there is no sunlight, these bacteria actually use chemicals coming out in the water from the mantle seeping out by these vents. You know, think of a vent next to a geyser, so it's close to the heat of the mantle coming up. And they use that to fix energy, right? And they can take in carbon dioxide from the water and make sugar, and they feed a whole community that's down there. So we have, if you look at eukaryotes, we have less diversity. These 2 exist in eukaryotes. But if we look at prokaryotes, we have all 4 possible combinations. So there's a lot more metabolic diversity in the prokaryotes than in the eukaryotes. So here's a nice example. So this would be a photoautotroph using light. Carbon source is usually carbon dioxide. Bicarbonate is how it's dissolved in water. So that'll be the ones in the ocean will be using that. And so there will be plants, but an example for prokaryotes would be cyanobacteria. Chemoautotrophs are those deep sea vent bacteria, right? Chemoheterotrophs, those would be animals and E. coli, for example. And then these are the ones that are a bit rarer, and those would be this unique to salt-loving prokaryotes and deep sea vent ocean ponds and hypersaline lakes like Mono Lake and the Dead Sea and things like that. Those would be your photoheterotrophs. Very, very general terms, looking at food and carbon. We have saprobes, and those are bacteria or fungi. You use these terms also for eukaryotes. And saprobics are breaking down, decomposing dead material. Parasites live on living hosts, sometimes internally, sometimes on the surface. And anything that can't be broken down, right, by these chemoheterotrophs, we refer to as non-biodegradable. So it includes, the most common ones are bacteria and fungi. And an example of something that's non-biodegradable would be plastic. Many plastics can't be broken down, and so they persist in the environment. Okay, the next one, so that's carbon and energy. The next one we're going to talk about in terms of elements, and we can do lots of them. And in ocean systems, if you take in oceanography, there are many, many more really important ones like iron and magnesium that aren't quite as significant on land. But we'll focus in on a very primary one, and that's nitrogen. And the reason nitrogen is so important is that nitrogen makes up part of the amino acid in a nucleotide, and amino acids put together make protein. And so you need nitrogen to make protein. Now I want to point out something that's kind of important, is that nitrogen, right, is taken up by plants in the on-land food chain, and plants make the protein. We're one step removed from that. We don't need nitrogen in our diet. We actually need to eat protein to turn it into our protein. But in either case, we need in the ecosystem nitrogen. And even organisms like plants, you might think about, well, plants doesn't have a lot of muscle on it. Why does it need that much protein? And you have to remember that chemical reactions in organisms are run by something called enzymes, and enzymes are protein. And so a plant that has all the sunlight and water, but has very pale yellow leaves and is not growing very fast, you have to add nitrogen, and that's the primary component in fertilizers to give to plants, because they're not making muscle, but they're making these enzymes to run all these other reactions. Right? Nitrogen is also a key ingredient in nucleic acids, and so for this nitrogenous bases, and so if it's really lacking, plants can't make protein, they can't even make DNA. And so nitrogen is important in your genes. So the nitrogen cycle is really important. We'll come and talk about that, and that's how it moves from one organism to another within the ecosystem. In terms of how organisms relate to it, nitrogen, they either can take it, just like when we're talking about carbon, some organisms can take nitrogen from the atmosphere and turn it into something useful in terms of something that living cells can use. If you're deficient in protein, you can't breathe in, deep breath in, 80% of the volume in your lungs is nitrogen gas. You can't do anything with it. Right? But luckily for us, there are a few bacteria that can. And they do something called fixing. And in this case, it doesn't mean it's broken. We can go back and call photosynthesis carbon fixation. It was taking carbon from the atmosphere and putting it into a bioavailable fixed, like solid compound. You use this term sometimes in, like, if you're making a plastic mold, you put in the liquid and you add a fixative, and what it does is it turns the liquid into a solid. Right? In that sense, it's fixing. So it's taking nitrogen from the atmosphere and turning it into ammonia that then can be used by the individual cell to make protein, or taken up by plants and used by protein. And so nitrogen-fixing bacteria in the soil are really important for soil fertility and keeping plants healthy. Nitrifying bacteria can take this. Remember how we talked about bacteria can be in biofilms? So if there's extra ammonia forming in the soil, there are other ones that can take it and convert it to nitrites and then nitrates. When we get to water treatment in the lab, we'll be talking about wastewater treatment, and we'll do a virtual tour of a wastewater treatment plant. Getting rid of ammonia and turning it into nitrites and nitrates, all of them can be contaminants in water, and we have to remove them. Right? Okay, and lastly, just somewhat rare, but there was a bacteria in the soil that when the nitrites and nitrates are in high abundance, they can actually release it back as nitrogen back to the atmosphere. This is from the atmosphere. So this is taking it in. And so if we look at our nitrogen cycle, right, here's nitrogen in the atmosphere. Then ones that are fixing it are taking the nitrogen in and converting it to ammonia. Then your nitrifying bacteria are the ones that would convert the ammonia to nitrites and nitrates. And your denitrifying bacteria are the ones that are putting it back into the atmosphere. There's another term here I want you to take a look at, and this is called assimilation. And this is taking in the element and making compounds. So in this case, the plant will take up ammonia and then turn it into protein. And so here the ammonia is taken in, assimilated into the plant. This looks like a little squash plant. All right, and so it's making a zucchini, and then this little rat is eating the zucchini. So the nitrogen is getting into the protein of the zucchini, then it's getting the protein of the rat, and then the rat could die, and then that will be released back to the soil. The plant parts die, is released back to the soil, or it pees or poops and gets back into the soil. So it makes what we call a cycle. And in a cycle, it's going from one form to to the next, and back. And so we have nitrogen in the atmosphere, getting into ammonia, getting into nitrite and nitrates, right? And then getting pushed into protein, and then broken down and back to the atmosphere. So it's making a cycle is what we call it. So that's why it's the nitrogen cycle. We'll come back to this over the semester quite a few times. We'll be talking about it in our first labs when we're looking at Anabaena, right, which is the type of cyanobacteria which do photosynthesis. And so this is as complex as prokaryotes get in that we have three separate cell types. They're not showing you the aconites here, right? So we have the vegetative cells. They just grow. But what the heterocyst does is it can take nitrogen gas in the water, convert it into ammonia, and then all the cells can use it to make their enzymes. And so you can get pools of water on granite rock up in the Sierras, and you think there's no minerals there. How are they going to grow? But they can take the nitrogen from the atmosphere and use it as an element to make their proteins. And so there's some of the most independent organisms on Earth because all they need is water, sunlight, and then they can make most of their compounds. So this is Anabaena, one of the ones we'll look at in our bacteria lab. Just showing the role plants need nitrogen, as I mentioned, to make ammonia. There are other strains, and this is really important in organic farming, and this case is actually showing you a forestry project. And it's showing you that in this case, it's not bacteria fixing nitrogen, but the growth of these types of bacteria cause nitrogen that's been stored up in the rocks and the soil to be released. And so the growth of these ones these strains of bacteria are actually making the soil nitrogen in the soil more available to the plant versus ones without it. So that's a little bit different. Actually, this one's not nitrogen. This is actually potassium. So this is showing you the release of potassium. And so bacteria obviously are important for nitrogen. Here it's showing you for potassium, and I didn't include the potassium cycle. It's also important for magnesium and iron, lots of them, right? So bacteria, really, really important for soil fertility. Okay, so that was nitrogen. So we've done energy and carbon, right? We had our photoautotrophs, things like that. We have our nitrogen cycle. Probably the most important one you want to remember for this stage is the nitrogen-fixing bacteria. You want to remember those. These other ones we'll talk a little bit about when we get to the water treatment plant and we get to the nitrogen cycle later on. But nitrogen-fixing bacteria are crucially important. Actually, when they first started doing genetic engineering decades ago, one of the first things they promised that was going to change life for us so much is they were going to incorporate nitrogen-fixing bacteria into plants that don't have them. Some plants, like when we get to the legumes and look at root anatomy, actually feed and make a chamber in their root and feed the nitrogen-fixing bacteria, so they have a built-in fertilizer factory. And they were going to put those into like tomato crops, plants and corn crops and things like that, so that we wouldn't need fertilizer. And so far, they've gotten into 0 plants. It's turned out to be really difficult. But that's just showing you they're really important. So I want to focus in on that for that. Okay, for oxygen, these are terms you probably already know of, and that's aerobic versus anaerobic. Aerobic uses oxygen. Anaerobic doesn't use oxygen. But the term you may not know, obligate versus facultative. Obligate sounds like it means, and that means it must have. So an obligate aerobe is like us. If we run out of oxygen, we're dead. End of story. Right? Anaerobes are ones that do not use oxygen. And ones that are obligate anaerobes cannot live in the presence of oxygen. And you might wonder, where on earth can you have areas without oxygen? Well, it turns out there's lots of areas because the obligate anaerobes in many cases grow very fast. And one of the things when we get to talk about wastewater treatment, we're going to be talking about BOD, biological oxygen demand. And what will happen is the aerobes will grow so fast, they run out of oxygen before they run out of food. For example, if we were having a party in a room and there's 30 people in there and there's a big table and whatever reason something happened and all the doors and windows got shut and it was completely sealed out, we would run out of oxygen very fast rather than food that's all distributed. It would take us days and days and days or weeks to run out, to die of starvation, but only minutes to run out of oxygen. And so where those happen, like in water systems, you can get anaerobic conditions forming very rapidly. Marshy soil is the same thing that happens there, that there's so much food that the aerobes use up all the oxygen. Intestines of vertebrates in particular, they run out of oxygen. And so early on, the aerobic bacteria use all the oxygen. There's no internal blood supply to the inside of the intestine. And so most of it's anaerobic. And so you can get lots of obligate anaerobes there. Only other term I want to add is facultative. And what facultative means is they can tolerate that condition. And so, for example, yeasts are a good example of that. And so I like to make wine and beer at home. And so I'll make my grape juice and I'll add the yeast to it, right? And then afterwards, I tap it and have a little bubbler with water so that the pressure, the carbon dioxide can get out, but oxygen can't get in. And so what will happen is that will form it to be, over time, the yeast in there use up all the oxygen. It becomes anaerobic. They still survive, but they survive doing a process called fermentation. And that's where you actually generate the carbon dioxide and the alcohol that's formed from that fermentation. But that's facultative. They would prefer, that's why I cap the bottle to keep the oxygen out. Because if they're growing with the oxygen, they wouldn't be doing the fermentation and you wouldn't get the carbon dioxide and the alcohol for making like champagne, all right? But they don't die, so that's the facultative. Part to remember is they can think of facultative and mean tolerant, right? Yeasts aren't facultative aerobes, meaning they can tolerate oxygen. That's not the case. They're facultative anaerobes. They can tolerate and still continue to survive, but not thrive in terms of the numbers of the cells. They can tolerate anaerobic conditions. So that's why we have a facultative anaerobe. Where these are taking place is in terms of the metabolism for both photosynthesis and for nitrogen metabolism, and for the oxygen is related to respiration. They will be taking place on specialized membranes. And I went over how they have specialized membranes, that they're an extension of the cell membrane folded in. So I just want to point that out. And so these would be the thylakoid membranes in a cyanobacteria. And these would be respiratory membranes. And so these are relating to both the use of oxygen and in here carbon dioxide, but also if I put up Nitrobacter for nitrogen. So these processes that are taking place, all are taking place on these specialized internal membranes. Not separate organelles, because these are invaginations, folding ends of the cell membrane, but still we do have special membrane regions within the cell. Okay, so looking at this, we talked about metabolism. This is an example of evolution. So we talked earlier on about how evolution works. This would be microevolution. We're not at the point where we're getting to new species. But remember, evolution takes time. And the thing about bacteria in particular is their lifespans are quite short. And they can replicate through binary fission in ideal conditions in lab every 20 minutes. So you can get multiple generations. So what they did here is they had a low sugar environment, and this is the original starting population. And they have the growth rate. So 1 is growing as fast as the original population. So they didn't have to have enough of this food source that they survive, but they're not thriving on it. And then what they did is, look, this is thousands of generations. Think, how long would it take to get 5,000 human generations? Well, we're talking about hundreds of thousands of years, right? Or tens of thousands of years to get 5,000 generations, right? So it takes a long time. But in bacterial populations, it's very easy. They can do this over a couple of years. And so all they would do is they started with their original population, and then they would transfer what they call an aliquot, a small little amount, and put it over into a new tube without any bacteria, sterile media, but it's the same media. And they're showing that over time, when they do that, right, they can actually see this population adapting and becoming better and better and more efficient at breaking down this unusual media. So there's a strong selection pressure for these different types of metabolism. A couple things before I move on to viral particles is we can use this ability, this metabolism that they have in plasmids, and they've been engineered now to take advantage of it. Some is simply selecting what they already do. Others are engineering and putting genes in. And we call this bioremediation. And that's using organisms to mitigate environmental concerns. And so here they're showing you after an oil spill, they actually can go with bacteria and spread it on the oil-soaked rocks. And they've engineered now bacteria that can start breaking apart the oil droplets. They don't really digest the oil, but they actually kind of like act as a detergent and break it to smaller droplets that then can disperse it into water to help get it into the water column, and then other things to start breaking it down. They've engineered bacteria that are good at accumulating heavy metals, and so they can use that to absorb metals out of contaminated soil by washing them with water and using the bacteria. And so this is called bioremediation. There's several bacteria that they're engineering to be able to break down. They've engineered several strains of bacteria that are fairly good at breaking down different types of plastic. And they're hoping to introduce them and use them to get rid of some of the plastics in the ocean. Okay, two more things I want to go over. One is the domains, the phylogeny, and then we'll talk about other life particles. And so here's two diagrams, one going in different directions, but I want you to notice in both of them, we have our three separate domains. And if you look at time, right, this is our origin here. So this is older. This is more recent. So now today we have three branches. And so you have the universal common ancestor. But then you'll see it branches off between the bacteria and the archaea, and that the eukaryotes and the archaea have their common ancestor as well. And so I want to point out that the universal ancestor, when it's at the cell level, this would be prokaryote. Because archaea are still prokaryote. And then we have the eukaryotes getting that specialization of the membranes and internal organelles. So let's take a look at why do we think this, right? And so if we look at this, I told you early on the first part of the lecture that in my diagram, when I drew it, you couldn't tell the difference between an archaea and a bacteria. And so we have our three domains. We have eukarya, archaea, and bacteria. Should probably move me out of the way there. And so if we just focus in on those three, notice there is a huge diversity. All of these things we're not going over. We'll go over gram-positive and cyanobacteria as particular types in the lab. We're not going to say anything about the archaea or any of these other types of bacteria. So there's a huge diversity there. And notice all of eukaryotes, all the plants, animals, and just one little line. So it's showing you there's a huge amount of diversity we're not going over. Looking at them, I want to point out at this little spot here, what does the common ancestor with eukarya and archaea, and why do we think that? So why not draw this differently? Why isn't this drawn like this? So you have the prokaryotes over there and the eukaryotes. So we'd have bacteria and archaea as a prokaryote branch, and then the eukaryotes over here as a eukaryote branch. Why isn't it drawn like that? So what evidence do we have? And so if we look, this is the way it's simplifying as best I can to show the relationship that I want to look at. We want to say, why is the domain archaea and eukarya share a common ancestor separate from the bacteria? And so if we look at this chart, it's giving us this information. And so what we want to look at, and I'll start with red, is we'll have what's coming from this common ancestor. And let's say this trait still go into the bacteria. So what do these have in common? And so They have prokaryote traits, so they don't have a nuclear envelope, they don't have membrane-bound organelles. Look here, they have the single chromosome. And so these are things that they get from being or inheriting from the common ancestor and being prokaryote. Okay, then if we look, we have a couple things that have come about in the archaea since they branched off from a eukaryote. So they're unique to archaea. And so if we look along, they can grow at very high temperatures. That's unique to the archaea. And so another one up here. One kinds of and in their membrane, they have some unique hydrocarbons. So the membrane lipids and the growth and temperature, that's some things that have happened since the common ancestor. And then what are unique and have happened from here and have passed on to the eukaryotes and the archaea is that they have RNA polymerase. How they start, this is a very 130 thing, but how something with their protein synthesis. Their gene structure having introns, so parts of the RNA are excised out. The resistance to certain antibiotics. Their DNA has little bits of protein in its structure called histones. And the lack of peptide and glycan, or that was probably something that was here and lost. So those are things that join the eukarya and the archaea together. And then, of course, in the end, what haven't I used? Purple. We have things that are new to the eukaryotes since they branched off from the archaea. So looking at it, the things in blue tell us that we know that the archaea are more closely related to eukaryotes than they are to other bacteria, the other prokaryotes being bacteria. And if you look at it, it's just not one, and you have to think, well, if we were to draw it, so we have archaea and bacteria versus eukarya, which is not correct. Or we have bacteria, then branches over here with the archaea and the eukarya. What would explain it? So if you look, if this were the case, we would have, my pen back. We would have 1, 2, 3, 4, 5, 6 traits emerging separately, because they're not from a common ancestor in this scenario, and with having the same end result. And if you think about evolution, mutations come about by random. Or we could have these 6 traits forming here and passed on to both of these populations. So which makes more sense? The fact that we have a common ancestor deriving these 6 traits and then they specialize into the archaea and the eukarya, or independently we get the same 6 traits coming about. And so the chances of those are very low. So that's why we know that this is the case and that didn't happen. And so these things give us the evidence, right? Lots of them to remember, but I would just focus in on remembering, if I would ask you on an exam evidence, the RNA polymerase is a big one. And that would help you understand how they relate. It's a very significant feature. I also have the peptidoglycan in the cell wall. It's easy to remember because it's a feature you have to remember for the bacteria. So if I were to ask you 2 of those things, those are 2 good ones and easy ones to remember. What RNA polymerase is a very key enzyme in the protein. So going from DNA to making a protein, you have to take the DNA and make it into RNA, and then RNA turns into the code that the ribosome uses. So that's a lot of 130 stuff. But just remember, it's a very, very key enzyme and highly conserved, meaning it wouldn't change very much without losing its functionality. And the fact that it's the same in these 2 branches really gives strong evidence of common ancestor, along with the other ones. Okay, so that's how we know it's this scenario and not that scenario. So don't remember that. And so remember the evidence that goes along here. Remember, red is from ancestor, and archaea still have it because they're still prokaryote, but eukaryote has lost it. Yellow is stuff that is specialized since they've separated out, so that doesn't give us evidence either way. It doesn't say that they're more closely related. Bacteria don't have it. Eukarya don't have it, so the yellow don't give us any information on it. In this case, it's the blue. The things that these two arms share that aren't in the bacteria, they only can be explained as coming from a common ancestor. The purple appears the same as the yellow. Those are things that are new since the common ancestor, and they don't tell us any information because the purple is not in the archaea or the bacteria. No information about a common ancestor. The yellow traits aren't in a eukarya or a bacteria. No information about a common ancestor. Right? So it's these blue traits that are telling us that. So putting that together gives us pretty strong evidence that we can say a little bit about the origins. Okay, if we know a little bit going here, these all come back to a common ancestor. We'll come back to this. You don't have to memorize this, but I'll just present it so while we're thinking about the common ancestor, things that all life, whether you're a bacteria, an archaea, or a eukaryote, they have the same DNA system. So that's why you can take a gene from a bacteria, as I mentioned before, and put it into corn. They run using very key enzymes in these processes. RNA polymerase is one that I just mentioned. When we get to metabolism, they all do glycolysis as the first stage for making ATP. They all have phospholipid bilayer as at least initially a cell membrane, and then eukaryotes multiple other interior membranes. So those are some of the things that go all the way back. Okay, archaea things to remember, they lack peptidoglycan, right? They're RNA polymerase. What was on that chart, if we actually look at the ribosome structure, there are features of the ribosomes in the archaea that give rise to show that the ones in the eukaryotes have a common ancestor with them. So those are some of the key features you want to remember. If I were to ask you, why do we think eukaryotes are more closely related to archaea? Or why archaea are more closely related to eukaryotes than bacteria? And why we have those three domains. Okay, I want to describe the archaea a little bit. They're probably best known for an unusual feature that they've adapted to, and they were first known as extremophiles. So they can live in very extreme environments. And so extreme halophiles, halo means salt. So they can grow in environments that are much saltier than ocean water, like the Dead Sea, or in California, Mono Lake on the other side of the Sierras. They can exist in environments that are above boiling in temperature. And so they can actually be preserved in pressure cookers and then released in the environment. And so we find them in thermal hot springs. And so the water isn't boiling when it comes out, but is nearly boiling. But they can survive in boiling water. They don't die. So they're super adapted to very high temperatures. And they also can produce methane. And so they're anaerobic. They live in the intestines of vertebrates, and they can take some carbon dioxide that's being produced by other bacteria in the gut, and they reduce it to methane. And that's an end product. And we'll talk a little bit about that when we get to ecology at the end, because methane isn't very abundant, but it's much stronger, about 80 times more efficient as a greenhouse gas related to global warming. And so for fermentation in ruminants like cows and what they feed cows. Those are methanogenic, and those are archaea. Those are the ones that make you fart. All right, so methanogenic archaea make you fart, not bacteria. And so here's so this will be an extreme thermophile. So this water is at higher than boiling because it's under pressure. It's coming out and constantly. And so constantly bathing these rocks. Of course, it cools a little in the air, but still this is near boiling, 60 to 80 degrees Celsius. And they're growing there. They're also photosynthetic. So you can see the colors that we're getting here. These are photosynthetic, but they're in different salt ponds. And so this is a picture taken down. So these would be the extreme halophiles. This picture is taken down by San Jose. These ponds, I think, are now no longer used for this. But what they would do is they let the bay water into the pond, like one right here, it was about the same color, and let it evaporate. Then they let more water in, so each time they flood it with salt water and it evaporates out, they're concentrating the amount of salt, right? Each time they add in more salt and concentrating. And you can see as they do that, the water, as it comes in, is dissolving the salt that was left from the last time. So water comes in the first time, has all the salt, same amount are in. Now it has twice the amount of salt as seawater, and then it can double again and double again. And you see these different colors, and those are different species of archaea that can survive on the different levels of salt that they're put in the ponds. All of these are photosynthetic. They're doing photosynthesis. And you learn about other types of photosynthesis when you take a microbiology class. But I just want to point out they're not green. They're not using chlorophyll. They are not generating oxygen. So some of the things that we associate with photosynthesis, we're really thinking about plants and cyanobacteria when they're doing that. So this is other types of photosynthesis. Don't have to be green to be photosynthetic. These are archaea, extreme heat, extreme salt. Here's hot springs around, I think this was Yellowstone, looking off towards, anyway, so this is the one that's in the textbook. The other ones are different colors, you can see. Okay, I want to switch over and talk about some biomolecules, and they're related to diseases. And so when we get to disease, we have two terms we use. One's pathogenic. They invade and attack a host. They get you sick, right? COVID-19, pathogenic. Opportunistic, or organisms that are on your body or there already and usually aren't causing any problem unless your body starts getting stressed out. And so what can happen there is they're in a very minor amount, and the concentrations that are there, bacteria get you sick by producing toxins primarily. And so the low levels there, they're not enough of a toxin to cause any problem. But let's say you take an antibiotic and it knocks out one suite of bacteria, but the other ones survive and they start taking over, and so there is an opportunity for them. And so when your body's stressed by one thing, it takes over. And so lack of sleep, being sick by a pathogenic virus or other bacteria stresses your body, your immune system's not working as well. And sometimes opportunistic infections start spreading out because your body's so stressed it can't keep them under control. And other role that bacteria in particular plays a defensive role. So you have Staphylococcus all over your skin, and the presence of the Staphylococcus bacteria prevents other bacteria that could be pathogenic from causing your sickness. And so this coating of the bacteria on your skin is actually defending you. So you can actually think of bacteria as defensive agents in some instances. Okay, and so bacteria, when you think about it, how can a bacteria get you sick? They don't have a mouth, they can't chomp on you. And so what bacteria do is they produce as a waste product or as part of their membrane or cell wall toxins, particles that cause your own cells to die is primarily what's happening. And so you may hear of something called food poisoning, and that's when bacteria grows in the food, and it's literally a poisoning event. Often it's not the bacteria themselves that are getting you sick, but the waste products that they're doing. Every living organism takes in metabolism. It takes in things. It recombines them, breaks them down, makes new things, and excretes things. And so if the toxin is secreted out, it's an exotoxin. That's usually what's happening in food poisoning. You leave potato salad out. Bacteria can divide every 20 minutes. So you leave it out in the warm weather and it gets warm. It could, even for a couple hours, the number of bacteria go way up. They produce the toxin. So even if you took that that potato salad and say, I'm going to put it in the microwave oven and cook it to sterilize it, it doesn't matter. The bacteria have already produced the toxin and it'll get you sick. And that's usually why you get food poisoning and it's so quick. You get the toxin, you're exposed to it, your body gets rid of everything, you feel fine, you got rid of the toxin. Endotoxins are ones when you need the bacteria present themselves and it's on the cell wall. Salmonella is one of these. And so as salmonella grows, it's the number of salmonella cells that you're exposed to that causes the toxic event. And here's a disease called Lyme disease that's transferred by bacteria, in this case by a tick, and has the bacteria in its saliva when it bites you and you get the disease out here. Lyme disease, when it's treated early, can be treated with antibiotics. Otherwise, the bacteria can actually get into your nervous system, and that's when it's really hard to treat because antibiotics have a hard time getting into your nervous system. And sometimes it doesn't make a rash at all. So that's an example of a pathogenic bacteria. In this case, it's actually producing an exotoxin. Okay, I want to finish up with things that aren't in the textbook few definitions and just examples of other things that are related to. They're not on our chart. They're not in a domain because they're not alive. And so the three general ones, the most important ones that we hear a lot about are viruses. They're referred to as particles because they're not a cell. They don't have their own metabolism, right? A virus by itself is inert. It needs a cell. It needs to infect a cell. And so COVID-19, if you cough and it goes out in the environment, they talk about how long it persists in the environment, and it'll dry up, it'll die, and then, it's not alive, but it becomes inactive, it'll break apart. It needs to be in a living cell. And so things that are caused by viruses, COVID-19 obviously, flus, all the types of flus, herpes can be as simple as a cold sore to other infections, HIV is the virus causing disease AIDS. And what a virus does is a virus has to infect a cell. It has a protein coat on the outside in DNA. Sometimes there's a few little enzymes in there. And so it will go into your cell. It'll inject its DNA. Your cell will make copies of the DNA. And the DNA carries the information to make the proteins, to make the coat. And then it will secrete out more viral particles. And so, and sometimes this process causes the cell to lyse. And so you get these blisters of the infected cells dying. Other times they can just remain there, excreting the cells, the virus particles from the cell. And so to get over a viral infection, you have to, trying to get this out of the way here, you have to get rid of the cells that are actually producing the viruses. And so it depends on what part of the body they infect and how to get rid of them. Your immune system uses T cells to do that. And what a vaccine does, well, let's get into more immunology, but it would prevent this from happening. Okay, the next one we want to go over are viroids and prions. Viroids is one main disease spreading through the tropics, and it's affecting coconut trees. And basically what a viroid is, it's like a virus, but it doesn't have a protein coat. It just has the naked RNA. And so the naked RNA gets spread from cell to cell. The cells actually reproduce more of the RNA and spread it, but in the process it infects cells and kills them. And so it's spreading all throughout the tropics. There's a third type, and this one's interesting. This is actually not involving DNA directly, and not even directly involving cells, and this is called prions. And what happens in a prion is you have a mutation forms a different form of a protein, a common protein, in this case one that's in the brain. And when the prion protein touches the normal protein, it causes it to change its shape. And proteins have a 3D structure. There's a particular shape that makes them work. And then what happens is it inactivates all of them. And then what happens is that the brain protein literally stops its function in the brain, and you end up with what's called mad cow disease, encephalitis. And it's the only known confirmed case of prions. There may be other ones out there. But I wanted to throw these out there. These are other types of things you may hear about. I want to point out they are biological. They're made by living cells. They are not cells themselves. And if you look at these processes, the way they get reproduced, passive tense, the virus doesn't reproduce, the cell reproduces the virus. It's messy, right? Notice this is happening outside of the nucleus. And so viruses mutate a lot. It's a very messy process. And so they can evolve. So these things are still evolving, right? Even though they're not cells, they're still under there's a selection pressure for viruses that are more effective or less pathogenic. And so those genes get passed on. So they do evolve, but they aren't cells. They're not technically alive. Lastly, 4 terms you may hear a lot of and I want to go over. And so this has to do with disease, prevention and protection from diseases. And so we have disinfectants and sterilizers. These are things like bleach and high heat, and they will kill all the viruses, all the pathogenic, I didn't go to eukaryote cells that could be pathogenic, like the amoeba, dysentery. So they'll kill all those cells and viruses and prions and viroids, right? But the problem is they're too tough to be used on living tissues. So you could gargle with bleach, and it would do a good job at killing bacteria in your mouth, but it'd also destroy the cells in your mouth. So that's a disinfectant, a sterilizer. You use them on non-living surfaces. Antiseptics are safe to be used on the external part of your body, like rubbing alcohol, mouthwash, you gargle and you spit out, you don't swallow it, right? And so, and alcohol wipes. So those things can be used, but they can't be ingested. Then you have antibiotics and antiviral, these things that are safe enough to be ingested, but they are targeting either antibiotics or targeting bacterial infections. And antiviral medicines, like remdesivir that they're using for COVID-19, are targeting the process of the virus reproducing or the virus infecting cells. Right? Notice antibiotics are against bacteria. So for COVID-19, you don't be taking antibiotics. It's not going to help because it's not a bacterial infection. Same with the flu or a cold. And so remember how antibiotic resistance builds up? The more people taking antibiotics, the more they're going to become less useful. So if you don't really have a bacterial infection, you don't want to be using an antibiotic. And so it's hard for doctors because you go there and you have a cold or a flu, and we don't have a lot of available antiviral compounds. And so people will want something. And so they demand an antibiotic, but it's not going to help. Right? Sometimes they'll give it for a viral infection remember we talked about opportunistic. So if there's a chance you have a weakened immune system and you're fighting a cold or a flu and they're worried about the bacteria in your lungs like tuberculosis, they may give an older person antibiotics when they're getting a cold or a flu to prevent an opportunistic bacterial infection. But by themselves, they won't help with a viral infection. Okay, so that's finishing up our chapter 27 on prokaryotes. And so we had 3 spots for the chapter. Following this, we're going to have a quiz. So you want to review the 3 parts before you take the in-chapter quiz. We'll have a lab of going over and learning about cyanobacteria and different types and the Gram-Stain procedure, and that will be included in with the quiz. So you want to take some time to review those things. Okay, next up, and we'll be carrying on some of this information, we're going to be talking about eukaryotes. And so I'll come back to that whole clade and phylogeny of how eukaryotes are related to archaea, and we'll be drawing on a lot of things that we learned about photosynthesis in the cyanobacteria and respiration, aerobic respiration with E. coli and things like that when we get to the organelles and eukaryotes.